There are 11 people, scientists in my lab, working on complement-mediated renal diseases. To the best of my knowledge, that's the biggest group in North America. So what happens when you have blood drawn and it's sent to our lab? I thought I'd walk you through the process so that you know why sometimes it takes forever to get your results and what's happening to your blood and all the steps that are involved. So when your blood sample comes into the lab, it's given a unique identifier by Amy. It's logged into our system. We have a huge database. The DNA is extracted from the blood. Some of you send serum and some of you send plasma. There's testing done by a lot of people, and you can see the names there. And then interpreting the testing is not straightforward. It's not like, wow, we found something, this is what it means, we send you a result. We have weekly renal group meetings. We have probably 10, 15 emails from physicians per week around the United States and North America asking, what does this mean? What do I do for my patient? How do I interpret this data? What is the next step? And so we actually discuss this as a group so that we're all on the same page, so we know what, as a group, we're doing and what we need to report to the physicians and to you so that you're getting the best care and the best information possible. Because these tests are offered in a CLIA-certified lab, because they're clinically available, any physician anywhere in the United States, in the world for that matter, can order the tests. They can send us a blood sample and request the test, and they don't have to ask you as parents or patients permission to get the test, because it's like when you go to your doctor and if you have strep throat and they do a strep culture and they send it off, you don't sign a consent form to get a strep throat culture. However, after our renal group meeting, sometimes, and it's a lot, we actually don't know what's going on. The result is unusual, it's unexpected, we can't explain it. And those are our pearls. Those are not the things we think, oh wow, we can't figure this out, we're not going to study these anymore, it's just like way too hard. Those are the ones we want to collect, those are the ones we want to focus on, those are the ones we want to sink money into, and those are the samples, the patients, the families that we devote a significant amount of time to trying to figure things out, because if we can figure things out in these families, it has huge implications for the entire medical community, the atypical HUS community, and the world community of the complement-mediated renal diseases. And a perfect example is Bill's family. It also leads to better test development, so there's a significant energy devoted in the lab to actually developing new tests that are important to atypical HUS, and in particular to focus on biomarkers, and that's kind of a buzzword for everybody. And what is a biomarker? It's something in the blood or in the urine that we can monitor that gives us insight into what the disease is doing. That seems pretty straightforward. Why don't we do it? Well, the reason we don't do it is because we don't know what the biomarker is that we ought to be monitoring and looking at. So that's the catch-22. It's a great idea if we find the biomarker, and so part of the focus in the lab, the energy that the lab is devoting, is actually to identifying what biomarkers we should follow. Now, we don't have, there's not a dearth of biomarkers. There's tons and tons and tons of biomarkers. We can follow lots of things. The question is, which one or ones are the best biomarkers to follow, and how do we figure that out? And the way we figure that out is what Carla alluded to. We get phenotypic information, we get clinical course information from you as parents and patients about the disease progression in a longitudinal fashion over time in people with atypical HUS, and we look at biomarkers, how they fluctuate over time, not in an isolated way, but in the context of the clinical course over time, in the context of what the creatinine is doing over time, in the context of what the LDH is doing over time. So as a global, in a global context. And by doing that, by focusing on that, There are new genes that are discovered that are important in atypical HUS and new functional studies that are being made. And that provides us better insights, better tests that we will develop to help clinicians, better tests that will help patients and their families, and better parameters to follow so that we have a more sensitive and acute understanding of what the disease is doing and how it's affecting a given person who happens to have atypical HUS.